I guess it's about time to start, and I'm happy to see so many that actually managed to wake up after the lunch coma and go and listen to something completely different. Because I have to admit, my name is Malin, Stridh, by the way, and I feel like a fish out of water at this place. I'm a neuroscientist. I've been to talks today and yesterday, and I don't even understand the titles of them. And now I'm going to bring you into my world a bit. So I did a lot of academic research, 10 years after my PhD, then went on to the more company side, the dark side, as some scientists call it. And now I'm running a stem cell company in Netherlands. So I'm going to present half of this time, and the other half of the time is going to be Kostas, who will probably bring some more maybe related stuff to what this conference is about. So I just want to say, is there anyone doing anything remotely with biology here? Oh, one, yay, two, great. So for the rest of you, if I go too fast or if I use too many acronyms, and Anton, I know you know biology as well. So just stop me. I mean, feel free to, I mean, we're few enough that we can have some sort of discussion, actually, during the talk. But I'll get started right away. We are going to talk about AI and personalized medicine and stem cells with some applications in cancer detection. So first of all, I would like to say that medicine today is evolving fast. The buzzwords, I'm going to introduce two buzz terms, I should say. One is regenerative medicine, which is basically about creating new tissue, replacing old cells. I mean, one of the most perhaps intuitive things is to think about diabetes, where the pancreas is not, the cells that are producing insulin, the pancreas is dying. We can make new ones. So that's what that's one application of regenerative medicine. There is also tissue engineering. It's a field that requires the work of many different disciplines. So you have the, let's see if I get this one to work. No. Well, okay, go for the top. You can see the cell source. This is something we are talking about quite a bit. We need cells to replace cells with, of course. And here we have the iPSCs, which means induced pluripotent stem cells. So remember that term for later, because there's going to be a lot of that. We also have personalized medicine, which is the current strategy, or up to now, it has been one treatment fits all. Do you get it to work?
Yep.
Oh, well, maybe it was just me that didn't see it.
Well, it needs a little bit of time.
There it is. Yeah.
There it is, yeah. Okay.
Okay, so this is the traditional way of treating illnesses today. We have a drug, most often a small molecule, and we give it to one disease. It's aimed for one disease, and then you treat all people with that disease with the same treatment. It's kind of working so-and-so. Future medicine, here it says more personalized diagnostics. That's one part of it. But you can also go in and look at, okay, so this patient A has the genotype of a certain makeup, and then because of that, it has to have treatment C. So it really tailors the treatment to each individual, because we're all different, right? It's not like the lab animals that we test the treatments on, because they all have the same genomics. So we're not the same. A lot of diseases have a lot of different causes. So we need to tailor treatments to the individuals, and that's the buzzword of personalized medicine. So going back to the stem cells, I mean, there's several different types of stem cells. And the good thing with them is, as you can see here, we have the starting material. Here it's named iPSCs, which means induced pluripotent stem cells. It can be embryonic stem cells. I shouldn't say it can be adult stem cells, because then the possibilities are limited. But if you use the iPSCs, you can get all these different types of cells. So you can talk about the pancreatic endocrine cells. That will be the insulin producing. You can have neural cells. That's something that we're actually doing in my lab at the moment. And you have chondrocytes, for instance, which means that if you need to replace the cartilage in your knees, that's the cell type you would want to use. So it's a lot of possibilities with these cells. And we actually have differentiation protocols for most of these cell types. So the iPS, or stem cell promise, is really that you can not only just replace damaged tissue, you can also, if you look at, let's go through the entire process. So you have the patient down here. From them, you can either take skin sample, fibroblasts, you can use PBMCs, so a type of blood cells, and you can convert them through a process called reprogramming into stem cells. That means that you get stem cells specific to that particular individual, with whatever issues they're having or with whatever benefits they're having. So this is what you do here, and you can either use a viral transduction method, which means you send a virus to the cells that gives them the signals to turn back time, or turn back development actually, and go back into a more embryonic-like stage, where it can, it's kind of like, if I dare to use a computer-like comment, wipe out the hard drive. You take away all the old memory tracks and make a clean one that you can store anything on, more or less anything. So you can do that either by the virus, or you can use mRNA, which is a version we're doing, which is nice. You can control that a bit more, and you don't have to worry about viruses being still in the cells. And then you get these stem cells up here. If you want to do something, if you want to correct something, let's say you have a patient with a disease that is caused by one faulty gene, you can go through genome editing, and you can get edited iPSCs, corrected iPSCs if you're trying to fix something, right? Or you can use them for differentiation to whatever cell types you want. Here it's motor neurons or heart muscle cells, or the insulin-producing cells. You can also take the genome-edited ones and do the same. And then, of course, this is what we're thinking about, stem cell therapy. But what's interesting is also that you can use it for drug screening. So if you have patients with an interesting genotype, or you just want to create something that is more resembling the actual situation, you can use the stem cells for that purpose, to screen for drugs with a more normal, or I should say, actual disease phenotype. You can also do modeling to understand the disease. I mean, there might be something you don't know about how it works, and then you get the stem cells, you create models, and you can look at what's going on there. So it's kind of a really, it's been in biology for a long time, the hype. When it was discovered, the stem cells were discovered, and the process, it was like, this is a solve-all, cure-all. Yeah, but we're not doing it, right? We've known this. The process of reprogramming was discovered by Yamanaka, and he got the Nobel Prize in 2012. Am I saying it right, Kostas? It's so bad with numbers or years. Yep. But I think it was 2012.
Yes, that's about right.
And we still do not have any approved iPSC-based therapies. So why is that? I think there might be one or two close to approval, but it's not living up to the promise that we believed it would. So part of the reason is really biology. What we often do is that we take cells from one individual, keep them, and then we want to treat many people with this. That can be done if we do not think about proper regenerative medicine. That is, if we don't want the cells to stay in place. If we want to replace tissue, this is going to be problematic because of the immune response. So if you use cells from one person and transplant them into another one, you're going to have the same problem as when you do a heart transplant, which means the patient has to be under immune suppression. For a life-threatening condition, that can be motivated, but for most of them, where we want to use stem cells, it's not motivated. Not even for diabetics, right? Where you could potentially replace the insulin function. But you still can use just exogenous insulin, and then it's not not really motivated to have the immune suppression. But then we have this one, the autologous cell therapy, which means you take samples from the patient and fix them and create the tissue you need, and then you transplant it back. So this is kind of the holy grail, because there you would not have to worry about immune suppression, you would not have to worry about all of these complications that could arise with graft rejection, but also the actual immune reaction. So this is the way to go, and we know how to do it. The process is not different from this process, more than you have to do the full reprogramming creation of the stem cells for each patient. And that, of course, is expensive. So main bottleneck of creating treatments is actually the reprogramming process, and it's a manufacturing process problem. So that's kind of disappointing. I mean, the science is what you think would be difficult, and it is, but we know quite a bit about it. It's getting these cells in an economically viable fashion that is hindering us at the moment. And why is that? It's labor intensive. So my lab people has to be in nursing these cells twice a day, every day, when we are reprogramming. So that's labor intensive. It's hands-on, it's people that does this, which also makes it error prone. So the failure rate is quite high. We have a dependency on human experts, and Kostas will show you what it looks like on images, because what we do is that we look at the cells every day during the reprogramming process, and that takes around 2 to 3 weeks. And we evaluate which cells. So in the beginning, you have one sheet of skin cells. That's what we're using, fibroblasts. And they have a certain look. And then after a while, when you have few cells that are starting to be reprogrammed, it's approximately 5 to 10%, if we're lucky, of the cells that actually do form stem cells. They start to divide, and they form little colonies that has to be distinguished from the remaining fibroblasts. And this is something that is evaluated by human eye, which means the standardization is poor. I mean, even if you have stem cell experts that are all trained in a similar way, they will judge different colonies in different ways, which means standardization for clinical purposes, difficult. And also it's costly. And if we scale up, if we get approved therapies that needs this process, we don't have stem cell experts available that can do the scale up. So we have dependency on human experts. It's expensive. The reagents are really expensive. It's done in rather large scale formats. You do it in either 6-well plates. Well, yeah, that doesn't really say anything to most of you. But you use quite a lot of these reagents, and they cost a fortune. It's time consuming. So to generate one line, if I'm buying specifically a custom-made IPS line, it takes me approximately 6 to 8 months to receive it currently. It costs around, if I want it clinical grade, it costs around 500,000 to €1 million. One line. You're not really doing this for every single patient, right? Not yet. And the difficulty is to scale up and out. So these constraints are effectively blocking the development of therapies using autologous iPSCs today. So what we want to do is to solve that bottleneck of creating the startup material so that we can take it on to differentiation and ultimately treatment. So how do we do this? And I guess you can figure out that we have an idea of this since we're here. And it's a project that me and Kostas has, together with the University of Leiden, and it's funded by the EIC. So we've been at it for 1.5 years, well, 1 year and a little bit more right now. And the whole thing is that we're trying to do is to standardize it. We're trying to miniaturize it so that we reduce the cost of reagents. And the most important part, I would say, automation and AI. So this part here is going to replace all the human bias and expert evaluation. That's what we're hoping, at least. So this is our solution, and I think Kostas will tell you a bit more in detail about it. So we're creating, this is a really nice computer rendition of where we want to end up eventually. But we're doing a downscaling into a microfluidic system that will allow us to grow cells, very controlled, under a microscope that continuously images the cells, that will feed data into the AI system to evaluate which colonies are good ones, so that we take out the human factor completely. Well, not really completely, but you can see here, so we aim to take it down to 2 to 3 months instead of above 6 months. Reducing the cost by a factor of 10 is what we're hoping for, and it's actually quite possible, just judging from where we are right now. Reducing the manual steps, and then eventually getting a fully automated reprogramming solution, which of course then also will be much more standardized and much more reproducible, which ultimately will lead us to getting approval from the regulatory agencies much faster. It's a lot of ultimately and eventually and hopefully. So my company and my knowledge is within the cell culture, imaging technologies, partly microfluidics. We've done quite a bit. It's all pretty easy stuff. I mean, we just need to optimize and test and test and test. The AI part, computers for me should just work. I should not have to put any effort into it. Discussing AI solution, I mean, for me, it's been hard to even know which questions I can ask of the system and how we can develop it. That's why I have Kostas. So I think I should leave the floor. And I should say that Kostas has a team of developers in Athens and AI engineers that we are working closely together with. And that's kind of interesting. It poses a whole other set of
challenges.
Issues. Yes, challenges. Because I don't understand what his guys are saying. And they initially didn't understand what we were talking about either. So do you want to take over from here?
Yeah, sure.
And we can talk about the communication gap.
Okay, so hello from me as well. So I'm Kostas Vougas. I'm the CEO of TipMed, who is actually in partnership with me in that great project. Just a little bit of background for me. So I am a molecular biologist. I'm not a computer guy at all. So I started as a molecular biologist, and I started working in a research facility doing proteomics. Just to explain, proteomics is the science of actually studying proteins. Proteins are the little molecules inside our cells that are doing stuff. So I started working on proteomics, and since, proteins are thousands and thousands like genes, we ended up being big data producers, like genomics. So thousands, tens of thousands of genes, tens of thousands of proteins, big data. And then I did realize that the main bottleneck is the data and handling the data. So I made, let's say, a turn in what I was doing, and for the best part of the last 15 years, I have been working on bioinformatics, biostatistics, and eventually machine learning. And that is how TipMed, the company, became a reality, because, well, at some point I just needed to accelerate stuff from the academic world to actually make things, do things, and bring things to the market. Yeah. So, as Malin said, we are a very small team of developers, 3 machine learning engineers, and we internally are faced up with the communication gap. So when the developers and the machine learning engineers sit on the same table, they don't have a clue of what one is saying to another. So that is a big problem. And the problem becomes even bigger when we need to communicate with Malin's team of stem cell scientists and neurobiologists and these things. So it's a totally different universe. So we actually, and that is one of the main points of us being here, we do realize that one of the biggest bottlenecks in what we need to do is cooperate, learn to talk to one another, understand one another. And maybe so new specialties might be introduced, like translators, one translating the technical terms of one team to the other team, so that we can move forward together. Everything that I'm going to show you from here onwards has been made a reality by developers. So if we didn't have developers, if we didn't have machine learning engineers, nothing would be possible. Yeah. So just the first use case, which is actually what we are doing in the AI PSC project. As Malin said, we need to automate a microscope so that it can capture cells growing in microfluidic chambers. So this would be the image collection part. Then we would also need to do something called Z-stacking. So take lots of images in multiple focus planes and then actually combine them together to create super resolution images, super informative images, out of which we can actually perform our AI and the decision-making of whether a colony is good or bad. Hence, removing the human expert out of the loop and being able to rely on an AI system that can automatically, persistently, always in the same way, perform the decision-making of whether a colony of cells growing is good or not. So the first part is automation, the second part is Z-stacking, the third part is stitching. So we have big channels where cells are growing, so we actually need to stitch the images together and create big, deep zoom gigapixel images, more like the Google Maps thing that you know, that you have the city from far away and then you zoom in until you see people in cars. So it is something like that. Lots of information. And then, of course, scheduling, which is actually taking images, these types of images, across multiple points in time and analyze all this big thing, put all this input into the AI. So, okay, more or less, what I've said is depicted here in the slides. So actually taking lots and lots of pictures, Z-stacked, combine them all together, stitch them as one, so more like the panorama in your iPhones, yeah? And then, of course, repeat this process across time so that we can collect the data set. And to give you an idea of the result, this is what we start with. So this is a single Z-stack from the microscope that it's collecting. So we have lots and lots of Z-stacks. And here, after applying image analysis algorithms and super resolution algorithms, we get the final result where you can really see that we have super fine detail of what's happening within the live system. You can even see the cellular cytoskeleton, so the skeleton structure of the cells. So quite big detail, because we do know that the biggest amount of data and detail you give to the AI system, the better it will perform. After that, we had to create a system to manage this whole information. So we created a cloud-based annotation. How are we on time?
Oh, all good.
Okay. So we had to create an annotation platform, a collaboration platform for all scientists, experts, our machine learning engineers, to go into annotate cells, annotate colonies, so that we could actually create a data set to train the AI. So we created this system which is deployed on AWS, and it is an internal tool that we need to utilize. It has complex working pipelines through which we can have annotator roles and reviewer roles. We have developed systems of AI, augmented AI-assisted annotation, where we have models performing the first crude annotation, and then we have humans actually correcting the annotations of the AI. And this being an iterative process, which is called active learning, in every cycle, the AI learns from its previous mistakes. Yeah. So
and I can tell you it's a hard work doing all the annotations.
Yes,
it takes time. We will be very happy when it's done.
Yeah. So and of course made it as such so that it can integrate with other third-party tools that we might require and other computer environments. After we have this whole data, we have compiled our first data set, and how we do it is actually feeding this data, feeding images to our deep learning neural networks. And firstly, we detect whether we have a colony or not. So first we distinguish colony or non-colony, colony versus background. We also detect borders so that we can know exactly what the line is separating colonies from the background information. And then we segment the image. We put circles, we mark the colonies. After that, we need to decide if a colony is a good colony or a bad colony. So since we have kicked out the background, we just take the colonies. We utilize image analysis feature extraction. So we take specific characteristics of images from image analysis algorithms and pipelines. And we also utilize deep learning, self-trained, self-supervised deep learning encoders to take in the images and produce features. So translate the image into numbers. And after we do that, we combine and we feed all this information to the classifier so that we train to distinguish between good colonies and bad colonies. This step is very important because we can, by translating image into numbers, then we can actually integrate all sorts of information inside. So if we have an image and make it into a list of numbers, and we also have clinical characteristics, or we have information about gene expression, so gene expression levels, we can actually integrate all this and improve our performance of the task that we need to do. And so finally, we get the classification of whether our colonies are good colonies or bad colonies. This is a slide that, well, we won't stay here long. I just put it here so that you can see. I mean, we have some metrics here. It's segmentation metrics. Segmentation is how good we separate our colonies from the background. Classification is how good we distinguish between good or bad colonies. And what these metrics actually tell you is that we have reached expert performance. We have matched expert performance. An important aspect beyond that, further to that, actually is AI explainability. So AI is a black box that lots of things are happening in there that we don't understand. But if we try and understand how the decision-making is happening, then we can learn new things. So this is, okay, this is a slide where here you cannot read anything, but these are features. These are features. Some of them are features coming from image analysis. So the shape of the circle, the shape of the colony, and so on and so forth. There are some other ones, the short ones, are features coming from the deep learning encoder, so we don't know what they mean. But we see that there are certain features that contribute to the decision-making either towards the bad colony or towards towards the good colony or towards the bad colony. So this is an opportunity for us to learn new things, understand new things, and this is also very important for the whole thing of a regulatory clearance, because eventually we are creating a medical device, and we need to be able to demonstrate to the regulators that we demystify the black box, make it as opaque as possible.
I mean, that's super important, the regulatory part, but it's also quite interesting because you potentially generate new biological knowledge here. You get some indications from the images, because at this moment we only have image data, so morphological data, and then you can, if we get to the point where we figure out what it means, it can give us hints towards what we actually should look at, also in protein levels and in general.
Yeah. The beauty of it is that actually one part of the program that is in progress is actually integrating gene information into the images in the way that I've talked to you about before, and then we can actually feed the whole explainability. We can feed this information to this whole explainability pipeline, and so we can get associations of gene expression to morphology, and that would be super cool because nobody knows about these things. So, new knowledge generation.
And now you have to talk really fast.
And now I have to talk really fast. Okay, so this is a second use case. This is actually the first product that the company, DeepMed, managed to bring to market. It is a CE-marked product that solves a solution. So, second use case, again, unfeasible without developers. The problem that it resolves is very simple. Histopathology lies at the center of all diagnostic processes, especially oncology. 90% of all pipelines in oncology depend on histopathology. And there is the problem known as workforce crisis. The demand is rising, the requests are rising, and histopathologists are decreasing. So you get a problem of delays. You probably get 2-month delay in order to get your report. And because the histopathologist was under pressure, most probably your report is going to be not in the quality that it should. So we are talking about misdiagnosis here. Our solution, which was implemented through funding of the NHS, is a decision support system that detects metastasis on H&E-stained lymph nodes from patients with breast, colon, lung cancer, and melanoma. So actually helping histopathologists in staging very quickly. There are two pipelines on diagnosing cancer: the grading to understand how aggressive the tumor is, and the staging to understand how much it has spread across the body. So the first step into doing staging is to examine lymph nodes to see if it has gone to the lymph nodes, because this is the first stop before it metastasizes. So imagine that there are 200,000 slides a year generated in a histopathology lab, 20% of which are lymph nodes. So it's a very boring process for the doctors, very tiresome, and it's a very huge volume. So what we do here is identify the tumor with very high sensitivity and specificity and show it to them so that we can make them do their diagnosis faster and with greater accuracy. This was easier said than done, because in order to bring it to market, we had to do lots of stuff like that, well, for getting the CE mark, which is actually make it fast, which we have done through parallel processing, and we have enabled the system to live in the cloud or in the premises of the hospital. And if we have 10 images and 10 GPUs available, we have enablement. We have parallel processing enabled so that each GPU is number crunching one machine, one image. Sorry. We started the analysis time being 45 to 50 minutes, and now we have, with optimization processes from our developers, we have brought that down to less than 6.5 minutes. We had to add through SDK support for multiple formats. from vendors having scanners digitizing the digitizing the microscope, the microscope slides, and of course, nobody liked software that it's on its own, so we had to integrate into software already running in the hospital through their APIs. So it was a huge effort, which is currently ongoing. I won't go through the performance, which is very high. What I will say is that we are making doctors on average 2.2 times faster, and we can save thousands of euro per specialty, breast, colon, lung, and melanoma, when they're using the system. So faster and cheaper. This is a solution enabled by AI, developers, machine learning engineers brought to the hospitals. Right now, it is procured in the UK as we speak in more than 26 hospitals.
So could we say that they're actually augmenting biology?
Yeah. It would be augmenting biology. Future plans is to actually make it the first pan-cancer metastasis detection system in the world, detecting more than 15 metastatic cancer types. And having said that, because we haven't said anything about personalized medicine, you'll give me a couple of minutes just to show you a work we did a few years back, since it was actually the first published work that actually demonstrated that deep learning networks can be effectively utilized in personalized medicine in this use case by predicting response to therapy for oncology patients from models trained on cell lines, specifically on their baseline gene expression. Baseline gene expression means the status of the genes, the expression level of the genes before these cell lines received any therapy, any drug. So the idea is to have so we had a data set of around 1,000 cell lines. We knew their baseline gene expression, around 12,000 genes for each cell line. And then we had a large matrix of chemotherapy reagents that these cells were treated with. And having known their response through something that we call IC50. IC50 is the concentration of the drug that is required to kill half the cells. So if you have resistant cells, you need that concentration goes high. If you have very sensitive cells, you need a little bit of medicine, a little bit of drug to kill it, so the IC50 is low. We had this information, and we trained lots of different deep learning models to predict that IC50. And then what we did was we went to actual oncology cohorts, clinical trials, actual patients. This is a clinical trial with a chemotherapeutic reagent called bortezomib. This is data from patients treated with another drug called cisplatin from TCGA, the Cancer Genome Atlas, a big project in the US that actually collected tens of thousands of patients' data on that. And here is something quite exciting, a clinical trial performed by a biotech in MD Anderson Cancer Hospital, and all three of them were actually failed attempts to distinguish responders from non-responders. And what you can see actually here is that the deep learning models that were trained on cell lines could actually, with statistical significance, discriminate responders from non-responders. So these guys here, we knew that are non-responders, and indeed they present on average a largest IC50. So it means that it can understand, it can distinguish the non-responders from the responders by their baseline gene expression. So before getting the treatment, usually the problem is that the patient gets the treatment for 3 months with a dreadful quality of life, and after 3 months they go to be checked, and then they were told whether this worked or not. which is horrible and decreases chances of survival. And not only did it manage to predict the short-term response to therapy, but it managed to also distinguish long survivors from non-one. So overall survival, which is known not to be poorly connected with the short-term survival. So this was a first step demonstrating that these tools can effectively be used in our battle against cancer in a personalized way. I'm finishing with this very quickly. This is like the slide that I showed you before. I'm not going into detail. This is our attempt. So this work was published in Cell Reports. This is our attempt to decode the black box and understand what was happening in there. And we managed to identify cluster of genes that are specifically involved in the resistance or sensitivity of specific drugs through pathway enrichment analysis. So new knowledge was generated, and out of this knowledge, lots of grant applications were also generated in order to look for these new associations. And at this point, I will give the floor to Malin to finish it up, because we have taken more of your time. This is something interesting before going away. This is when I asked DALL-E from OpenAI to imagine the future of medicine. So I asked it, please imagine the future of medicine, and that's what it came up with. And Malin?
Yeah, do you want me to tell you about the future of medicine?
Yeah. Yeah, tell me about the future. Tell us about the future of medicine.
To really wrap it up quickly, I think it's fantastic that when you ask DALL-E to do this, it shows you what, at least to my interpretation, is a picture of regenerative medicine. We can see spare organs being depicted. We can see the human skeleton being shown where to put the new organs and everything. Just to really wrap it up, it's a super interesting time in biology, medicine, the interface between data science and machine learning and tissue engineering and nanomaterials. I mean, all this comes together and we will be able to do great things. But we need to figure out how to solve the communication gaps, which, I mean, for me, it's mostly bringing people together that really wants to do something. Oh, yeah.
Oh, shut up.
I'll be quick. I'm just going to tell you 2 projects that we're doing or trying to start up at the moment, just to show you that, at least to me, it feels like we're living in the science fiction part, or at least close to it. One thing is creating your own insulin-producing organoids. I mean, that's done. It's tested in animals. The procedures are ready to try in humans. Pretty easy, but we're talking about encapsulation with 3D printing and adding other compounds to make sure that you have blood vessels growing into the transplant. So it's kind of almost there, figuring out the last parts. But the most interesting part for me is the spinal cord project, the spinal cord injury project we do. There we have 3D printed collagen scaffolds that will be tailored to each individual patient's injury site. We will seed these scaffolds with neural stem cells derived from the patient's own cells. We are considering combining this to ensure that because we've got electrical components when we talk about nerve cells, we're putting in graphene nanoribbons that in animal studies have shown to link and help axons to grow together and for neurons to connect to each other. On a side note on the graphene part, we're also talking about how to make new neurons connect to artificial limbs. So we usually, a little bit jokingly, and I know it's not completely ethical correct to say, we were talking about making cyborgs. For me, this is super fascinating. I would not have thought 10 years ago that we will actually seriously discuss and start the work with these things. But that's where we are, but we need more data people. So I hope we have inspired some of you to think that biology is quite interesting. It's complex systems. If you want a challenge, you're more than welcome to come and join us. So with that, I think we've held you long enough. If there are questions, we'll be happy to answer them, but you might want to, yeah.